All right, guys, welcome to another bio review. Hopefully this time y'all don't fail the tests as badly. JK, I know y'all passed the last one, right? All right, so first thing we need to cover. This unit is all about genetics, right? All your DNA and how it is represented by, you know, your current physical traits. So first thing we need to cover is genotype versus phenotype. When concerning genotypes and phenotypes, what's the difference? Anybody know? Maybe I should stop asking questions because I know y'all aren't going to answer. Okay, so genotype in this case is the exact genes, right? And phenotype is physical traits. Now, physical traits isn't the best way to word it, but it makes sense for how you're going to think about it, right? It's what is going to be displayed or represented, right? And based on this, so genotype and phenotype got developed in Mendelian genetics by a guy named, Gregor Mendel. Mendelian genetics, Mendel, same difference, right? But he discovered the difference between the genotype and the phenotype by crossbreeding plants, right? And using genotype and phenotype, he was able to determine two different qualities of animals. You can either be homozygous or heterozygous. Right? And homozygous and heterozygous can be explained by alleles, right? Alleles are basically ways your DNA encodes your phenotype, right? Alleles, depending on what alleles are present, you will display different physical traits. Some alleles are dominant over others, right? Dominant alleles will show their traits always. Recessive alleles will only show their traits if there are no dominant alleles around. That part makes sense? I need a yes or no. Anybody?
Yes.
Okay, good. So homozygous, you are either dominant or recessive. I should use lowercase d's. And heterozygous, you are dominant and recessive, right? These concern genotype. Remember, homozygous and heterozygous concern genotype. Hang on, let me mute some things real quick. Give me 1 second. There we go. All right. Cool. So everybody understand that whole part, because this is going to be what we build everything off of. If you don't understand the difference between homozygous and heterozygous, you're going to have a bad time for the next hour.
Yes, we get it.
All right, cool. Let's keep going. So, one of the things in Mendelian genetics is the law of segregation, right? The law of segregation concerns chromosomes, right? The law of segregation basically states that the segregation of chromosomes during meiosis is so varied that the chances of having two similar offspring is very, very slim, right? So in order for you to understand that whole thing, we need to talk about meiosis, right? We've already covered mitosis last time. Mitosis is doing cell division. Meiosis is specifically for gametes, right? Your reproductive cells. So meiosis does the entire part of mitosis twice, basically, right? Your prophase, metaphase, telophase, anaphase all happens twice. So let's start. So say, for instance, I have a cell. This is a gamete cell, and it has two strands of DNA. I'll do red and blue. right? And red and blue are going to be replicated, right? Because before you can do cell division, you need to replicate your DNA. So red is going to get replicated, and blue is going to get replicated, right? So now that red and blue are replicated, I'm going to change the shape of it. Now, red and blue are now replicated, right? This part makes sense. Before you separate your DNA and stuff, you gotta replicate it. The next thing that needs to happen is analogous chromosomes must pair up. That's a lot of big words to say. Chromosomes with similar alleles need to pair up, right? Say, for instance, the red chromosome had the allele for hair type, right? Straight hair versus curly hair, right? And the blue allele also, and the blue chromosome also had the allele for hair type, being straight hair or curly hair, right? So these two would pair up together. I gotta, oh no. I'm just gonna erase it. So these two would actually pair up together and move, well, as one unit now, right? So I've got my red, and I've got my blue. And if you're confused as to when we learned this, this was in the activity, which is why we didn't learn it. Does this part make sense? We are talking about analogous chromosomes. Now you can replace the word analogous with similar and it'll still make sense, right? These two chromosomes have the similar alleles. Oh yeah, don't forget to put your name in chat, otherwise I won't give you credit. I have to mark you absent, and then that's a whole nother pain in the butt when your parents email me. So now that all the analogous chromosomes are paired up, we actually have a term for this little grouping. We call them, write this in it, tetrads. Tetrads and analogous chromosomes are the same thing. Why do we have so many words for similar things? Who knows? It'd be like that. Thank you to everybody who's putting their names in chat. I appreciate it. So now we need to do the next step, right? We've duplicated our DNA, we've paired up our analogous chromosomes, and we've made a tetrad, right? All of this happens in, hang on, wait, something weird is going on. All of this happens during, prophase 1. This is prophase 1, right? You duplicate your DNA, you have your analogous chromosomes, you pair them up, and now they are tetrads, right? So now we have prophase 1 done, right? Preparing for the first half of meiosis. The next part is what comes after prophase. Anybody?
Metaphase?
Yes, metaphase. We go on to metaphase 1. So let's erase all this stuff. Everybody got the analogous chromosomes or a tetrad part?
Yes.
All right, cool. I'm gonna erase this. So now let's move on to metaphase. Now in metaphase 1, well, we have this tetrad. What we're gonna do is move it into the middle. So let's do that. I'm just gonna shift the outside. know that it moves into the middle. Now it's in the middle, and we draw an imaginary line. Yay, it's in the middle. We're done with metaphase 1. Yes, that's all that really happens. Next thing we need to do is anaphase 1. What normally happens during anaphase? Nobody remembers? Well, in anaphase 1, yes, they separate, right? But instead of sister chromatids separating, the two analogous chromosomes are going to separate. So this is going to go this way, and this is going to go this way. Now, your body has a bunch of chromosomes, but we're only representing it with one because easier visuals. So as they move that way, we now have one red chromosome here and one blue chromosome here. And next thing we need to do is telophase 1. In telophase 1, same thing that happens in normal telophase. Right? Your cell membrane starts to do this. Notice they are beginning to separate, right? This is telophase 1. And at the end of it all, you get, hang on. Come on, sorry, trying to get back to the whiteboard. And at the end of it all, you get the end of meiosis 1, right? Which is just two separate cells. Completely separated. And they have their own little nucleus around them. But now you have the end of the first part, right? At the end of the first part, what you need to know, and this is the key part, you have now made two haploid cells. To be a haploid cell, you need to have half the DNA of the original. And this might be a little confusing because let me redraw our original. Put a nucleus in it this time because I forgot to do that earlier. Our original had, well, 2 strands of DNA, right? Sorry, I should have done the second one in blue. 2 strands of DNA. And imagine this is a human cell. So how many chromosomes would that be? 46? Yeah, that'd be 46, right? And if we're looking at these two, logically they should have 46 because the DNA was duplicated. Well, pay attention to the colors, right? Notice how this one only has red and this one only has blue. That means, since they only have one of the 2 types of DNA that the parent cell had, they're haploid cells. They technically only have half the DNA of the parent cell. So at the end of meiosis 1, aka the first half of meiosis, that is when you have haploid cells. From this point on, everything else is a haploid. The parent cell is a diploid, meaning it has, the correct amount of chromosomes for that specific organism. Everybody understand this key point, because if you don't understand this, it gets very confusing.
So it's not 46 and 2 haploid cells?
No, each of these 2 cells only has 23, even though the DNA was duplicated. Everybody understand that?
Yes.
Who's iPad? Someone just joined and they're named iPad.
Wait, Marcus, can you explain the diploid thing again one more time?
A diploid cell is a cell with the correct number of chromosomes for that specific organism. Like if you're a human, your diploid cells have 46 chromosomes. A haploid cell has half that number. So your gametes as a human have 23.
Thank you.
All right. Now that we're done with that part, let's go on to meiosis 2, aka the second half. So let me erase our parent cell because we don't need them anymore. So now what do? Well, we now enter prophase 2. In prophase 2, this time we don't need to replicate the DNA. Why? Because we have enough. And so the nucleus disappears at the start of prophase 2, and well, pretty much everything's ready. Next thing we need to do, metaphase 2. Metaphase means what? Audience participation, please. Metaphase means they're all going to move to the middle. So let me redraw everything to make this make more sense. I'm going to put one cell here, one cell here. Oh, they shouldn't open. I'm going to have, gotta draw this one sideways. So now they're all in the middle, or as close to the middle as I can get it because I'm freehanding all this stuff. Oh boy, I'm about to have a cell with a mutation. Anyway, so metaphase 2, your two separate chromosomes now line up in the middle. Next thing you do, anaphase 2. What happens in anaphase 2? The same thing that happened in anaphase 1. They're going to separate. So in anaphase 2, this sister chromatid is going to go this way, this sister chromatid is going to go this way, this sister chromatid is going to go this way, and this sister chromatid is going to go this way. So now what we have is an interior cell that looks like this. So now I have a sister chromatid here, a sister chromatid here, a sister chromatid here, and a sister chromatid here. Now we enter telophase 2. And I know I'm going kind of fast, but they're basically the same steps. In telophase 2, you see the pinching. And then at the very end of meiosis, aka meiosis 2, you have 4 distinct gamete cells. Notice at the end of meiosis, the whole process, I started with 1 diploid, and I have now made 4 haploids, each with half the original amount of chromosomes and each with very specific DNA in them. Now, let's go back to what we were talking earlier about the law of segregation. Notice how some of these have the same DNA, but others do not, right? And it is during the anaphase process when chromosomes are being separated and sister chromatids are being separated from each other that the law of segregation really comes into play. The law of segregation basically says, due to this separation and the random chances of how it'll go, it is very, very unlikely for two of your offspring to be the exact same. And this current picture we have doesn't represent it well, but imagine doing all that entire process with 46 different chromosomes. Basically how it goes. Anyway, everybody understand the whole meiosis process?
Yes.
All right, cool. Another thing you'll need to understand is independent assortment. Alleles of one gene separate independently of an allele of another gene. Just know alleles separate independently. That's it. Anyway, now let's get into fun parts. Everybody knows how to do a Punnett square, right? So I don't need to go over that.
Yes.
Yes? Okay, cool. Let's talk about, what is it? So what did I want to cover next? I wanted to cover, all right, let's do pedigree analysis. Yeah, I want to do some pedigree analysis?
Yes, please.
All right. I figured I wouldn't be able to do this because this was done on an SLO. I have a completely separate section of notes just for this. So when you're analyzing pedigrees, you need to understand that pedigrees are very confusing and that there are 5 situations you can always apply to a pedigree, right? So let's draw an example. Say this person has 2 kids, a boy and a girl, and this person marries. Okay, this is a very simple one, and let's start marking affected versus non-affected individuals. Remember, females are circles, males are squares, and affected individuals are colored in. So let's look at this. There are 5 situations you can always apply to a pedigree tree. Autosomal dominant, which I'm going to shorten to autosomal recessive, sex-linked dominant, sex-linked recessive, and Y-linked. So let's go over these in one at a time, right? Now, the only way you are going to be able to figure out which one works on a pedigree tree is to test them all. And it takes a while, but it's worth it. Now, let's go over the easiest one, Y-linked.
Wait, so I'm sorry. On the test, that's what we're gonna have to do, all of them.
Yes. Hopefully she doesn't put a question like that, but you need to know the method.
Okay, thank you.
Now, I'm going to teach it very slowly, but you got to learn how to speed run this. So, Y-linked. Y-linked is the easiest one. When it comes to Y-linked pedigree trees, only guys will be affected. Are there girls affected in this tree? Yes, there are. So it's not Y-linked. We can cross that one off right now. So now we need to test autosomal dominant, autosomal recessive, sex-linked dominant, and sex-linked recessive, right? On autosomal, anybody remember what autosomal means?
Wait,
Probably,
Mark,
yeah.
for the Y linked, I thought if it's like if the dad had it and then the son had it, it's Y linked.
Yeah, but also on Y-linked, only guys can have it. If a female has it, it's not Y-linked. Because remember, girls are XX. They don't have a Y. So, now that we got Y-linked off the table, anybody remember what autosomal means? I'm going to guess no, so I'm going to answer my own question here. Autosomal means not a sex gene. So, when we're considering autosomal dominant, I always label my dominant as capital A and my recessive as lowercase a because easier to see, right? So if we think it's autosomal dominant, that means this person, I'm going to use this person as an example, this person needs to have a dominant gene and a whatever gene. It doesn't matter. We're going to leave that blank, right? That also means that this person, since they're affected, remember, it's a dominant gene, that means they have to have a dominant and a blank gene. Does that make sense?
Yes.
All right. Now, we leave it blank because it doesn't really matter. Because remember, if the dominant gene is there, the dominant gene takes over. So now we fill in the blanks for all of these. What about the people who don't show them? Well, if they don't show them and it's a dominant gene, that means we can assume they are double recessive because, well, they're not showing the gene. They have to be recessive. So let's fill this all in. And so now we get to the best part, testing whether or not we are correct, right? Now that we've filled all of this in, let's take a look. Now, can these two parents have two offspring which are double recessive?
Yes.
Well, yes. Okay. That is the correct answer, but I will show you the way to test it out. If you see a dominant and a blank, assume the other one is recessive. And that's how you will be able to test it out. So now we draw our Punnett square and we see, aha, it is possible to get recessive children. So this does apply. So let me do this in green. So this child is good and this child is good. So now let's look at the other one, right? Can these two parents here have these three types of children? So now let's test it out. I draw my Punnett square, my dominant, and I'm going to assume my blank is recessive because that allows me to test for everything. Well, it looks like it is entirely possible. So it is possible. You can hypothesize that autosomal dominant is the type of gene that is connected here. And now comes the most painful part, because you have to test them all. We're not done. Now we got to go through autosomal recessive, sex-linked dominant, and sex-linked recessive. So let's test out autosomal recessive. And if you want to save yourself the work, I'm just going to shortcut this one, right? So if I think it is autosomal recessive, let's take a look. I need to relabel everything. Once again, if it is autosomal recessive, that means all of the affected people have double recessive alleles. So this person is double recessive, 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 and this person has a dominant gene because they are not affected. This person has a dominant gene because they're not affected, and this person has a dominant gene because they're not affected. Now, we have to test them all out one by one. Can these two parents have these two children? I'm just going to draw the Punnett square now because it is going to be the same for everybody. Can these two parents have these two children? Yes, they can. Can these two parents have these three children? Yes, they can. Which means it can also be autosomal recessive. And if that is confusing to you, don't worry, it makes perfect sense. Now, everybody got the autosomal ones down? Because those are easy. Everybody good?
Yes.
Yes.
Okay, good. Let's do sex-linked traits. So now, when we're considering sex-linked traits, we've already determined that it isn't Y-linked, because if it was Y-linked, only guys would have it. Now, we are looking at sex dominant versus sex recessive. When it's just these two, you need to remember they only affect X's, right? So in dominant traits, right, you also need to keep track of who is what gender, right? So instead of putting, because for autosomal dominant, I would normally put a blank here, I would instead now put X, capital A, because I think it's dominant. So one of their genes has to be affected, and the other one is blank, because it doesn't matter, there's a dominant gene there, right? Now, if I look at this other parent, they will have an X and a Y. The Y doesn't matter, but the X does. If the X, and I think it's dominant, is not presenting that gene, that means it has to be recessive. Same situation here. This is a guy. He's not showing the trait. Has to be recessive. This one is a girl, but they're not showing the trait, which means they have to be both recessive because I think it's a dominant trait. This male has an X with the affected gene. And all of his guy children, the same. And the girl is showing it, but the other one is blank. So now we draw a Punnett square. This time we're going to have to make two because there's two different types of parents. So we have a female with the dominant trait, and I'm going to assume the other blank is recessive, and a male with the recessive gene. So now I have so are these children possible? This one is possible, and this one is possible. So let's continue. Let's look at the other set of parents. This time we have a male with the affected gene, and a female with neither. Y'all understand everything up to here, right? I know I'm going kind of fast.
Yes.
Are these children possible? No. Look, there are no males with the affected gene, which means it is not a sexual dominant trait. Now we need to test sexual recessive. And gotta erase everything again. If any of you did the SLO, you would have experienced this pain already. I hope y'all did the SLO. So now let's do this again. She is showing the trait, and I think it's recessive, which means she has to be double recessive. He is not showing the trait, which means he has to have a dominant gene. He's also not showing the trait, has to have a dominant gene. She is not showing the trait, so she must have a dominant gene and an unknown gene. This guy shows the trait. He has a recessive gene. This guy also shows the trait, has a recessive gene. This person also has a trait, shows the recessive gene. And this female is also showing the gene. So there we have it. Now let's draw our Punnett squares. Starting with the first couple on the top, I have a female who is double recessive and a male who is affected. So drawing out our situation. Are these children possible? Yes, no, perhaps. I look at this, I see this. That is not possible. It is not a sexually recessive trait, which means this whole tree can either be autosomal dominant or autosomal recessive. And I figured it out because I tested every single one. Y'all understand how to do these trees now, right? Please, I need an answer.
Yes.
Yes? All right, good. So now that y'all have a decent understanding of this, let us continue. So that's how you do those. Remember, you have to test out all 5 scenarios every time. And by every time, I mean every time. So now that we're done with that, do y'all want to do dihybrid crosses? Yes?
Yes, please.
Yes, dihybrid crosses. Okay. So, say for instance, I have this. Say for instance, I have these 2 parents. And I need to figure out, well, how do I do this dihybrid cross, right? What are all the genotypes I'm going to have, or all the phenotypes I'm going to have, right? So let's say capital A is tall, and lowercase a equals short. Capital B equals, I don't know, let's do a plan thing. B equals big leaves. Lowercase b equals small leaf. And so the first thing I need to do is, well, I got to determine what are the ratios for all of the traits. And in order to do that, let's do something that she's definitely going to ask on the test. I'm going to ignore this other parent. Remember, when you produce gametes, they all separate into very specific traits, right? If I only had A, I would produce 2 gametes through meiosis, one that has a capital A, one that has a lowercase a. Does that make sense?
Yes.
Yes, good. So I need to determine what types of gametes I'm going to produce from this. In order to do that, well, you do the FOIL method, right? What kinds of gametes am I going to produce? So I'm going to produce a gamete with, I do A times B, so that's AB. I do capital A lowercase b. So this parent will produce 4 gametes. One with capital A, capital B, one with capital A, lowercase b, one with lowercase a, capital B, and one with lowercase a, lowercase b. Does that make sense? This will be relevant in a second. I will try to make it relevant so that y'all remember this. So remember these gametes because they are the same for both parents, right? So let's bring back our other parent. So when I'm trying to determine my ratios, I got to look at these gametes, right? And I got to put them in a cross, right? Why would I solve for these gametes otherwise? So when I do a dihybrid cross, you can do it the difficult way, which she should have shown us because it would explain why you need to do the FOIL method to get those gametes. When I do it like this, I put my gametes here. Do y'all see now why solving for gametes is relevant?
Yes.
Yes? Because, well, yeah, this is why. You cross the gametes between both parents. She should have shown this to you all because that would have made a lot more sense. But yeah, so when a question asks for the gametes, it's asking for what are you putting into these 2 rows. Right? Not whatever is in here. It's what you're putting into those rows. Hopefully that cleared up some confusion. So now, let's do it the easy way. And the easy way is, well, I can just make 2 separate Punnett squares. One for A, one for B. Right. And y'all know how to do this part, I hope. So I'm going to ask, what is the chances of me getting a short plant with small leaves? And I will fill this out slowly while y'all solve.
Do you want us to like say it or type it?
Type it out, say it. I will look at it either way. I'm not Simona's. I have chat open for a reason. Because I know most of y'all aren't comfortable speaking in front of your peers. All right, Casey got 1/8. Anybody else? We're looking for short with small leaves. Remember, short is lowercase a, small leaf is lowercase b. Oh, Casey says 1/16. She corrected herself. Anybody else? Or do all of you agree? All right. Well, I didn't hear anything from anybody, so let's see if any of you agree. So in this situation, I have 1/4 of them are going to be short because lowercase a is what I'm looking for. And I have 1/4 of these is going to be small leaves. I multiply them together. 1 times 1 equals 1. 4 times 4 is 16. Good job, Casey. You got it right. I wish I had a reward to give you, but you know, Corona. Not exactly safe. So now y'all know how to do that. Fairly easy, right? Right?
Mary, yes, thank you.
Yes. Okay. Let's do one more situation before. All right. Solve this one for tall with big leaves. And then, yeah, you know, I'll just make y'all do that much. I won't make y'all solve for gametes because y'all should know how to do that. So do A. Oh wow, this one's easy. Oh, yeah, I'm not going to give y'all the answer. All right, let me see them answers. I made your squares for you. Answers, anybody? One. You know what? Here, to stop confusing y'all, because I know this might be a bit confusing. Put it in percent. Give me a percentage. What are the chances?
I think I'm wrong, but 50%?
50%. Oh, that's an answer. Let's take a look if you're right.
Just
So
curious.
I'm looking for tall. Tall is capital A. I see 1, 2, 3, 4 out of 4 times, and I'm also looking for big, which is capital B. So I see 1, 2, 3, 4 because it's dominant, which equals 16 over 16, which is 100%.
Oh, just kidding. I typed that wrong in the calculator.
Ah, okay. That made sense. I was confused for a second there. All right, that's how y'all do dihybrids, right?
One wrong.
One would technically be correct. I would have written it as 16 over 16 though, because that's how she had us put it last time, which was weird. But one is correct. So now let's do what's another type of dominance? Let's talk about codominance. So codominance is basically when two things share dominance, right? You can be dominant in two forms. AB blood is technically codominant, right? So let's do blood types. Blood types are always fun, and that's the example she's going to use. So say, for instance, you have an AB blood type parent that and BO, right? O is recessive to both A and B, but A and B are codominant. So to explain that, let's draw a square. I have A and B O. So to explain, A is always O is always recessive to A and B. So in these two situations on the bottom, the child will either have A blood type or B blood type and carry the O gene, right? Whereas here, this person doesn't have, let me do it in a different color. This person doesn't have the O gene, but is still a B blood type because, well, they have both B alleles, right? And this person here is AB, which means they're going to have AB blood because they are codominant. They get to share the spotlight here, right? So for this situation, we would have a 1 in 4 chance of an AB kid. We would have a 1 in 4 chance of having an A blood type kid and a 1 in 2 chance of having a B blood type kid. Why? There's 2 Bs. Even though this one's a carrier, they represent the B blood type. Does that make sense?
Yes.
Yes? Okay, cool. So let's do another situation. I have O plus BO. Give me all of the phenotype ratios. It's only out of 4, so it shouldn't be too many. And remember, I am talking about phenotype ratios, so I want to know what their blood type is going to be. Oh, come on, undo, there we go. That's the square if you want to set it up like that. So when you're done, go ahead and say the answer or, just type it out in chat. All right, what are we getting in chat? 1/4 for all. Okay. 1, 1, 1, 1. Good ratio, good ratio. Okay, I got 2 answers in. I will solve this for you. So these 2 will cross, giving us an AB. These 2 will cross, giving us ABO. These 2 will cross, giving us an AO, and these 2 will cross, giving us OO. My dad messaging me concerned. So the chances of me having an AB kid here, 1 in 4. Chances of having a B kid, 1 in 4. Chances of having an A kid, 1 in 4. Chances of having an O kid, 1 in 4. Everything makes sense, right? Yes, yes, yes.
Yes.
Yes, good. Cool. Let's talk about incomplete dominance. So when I have, say, for instance, a plant with a a red or white flower, right? So we have red, red plus white, white, right? And in this situation, it is incomplete dominance. She'll tell you on the question if it's incomplete dominance, or you'll see what are the chances of getting a pink offspring. Well, I don't see pink on here, but I can assume that if I get a mix of red and white, it equals pink. Because here it is incomplete dominant. So let's draw our Punnett square. If I have a plant that is solid red, it will have both of the red alleles. If I have a plant that is solid white, it will have both of the white alleles. And I know that in order to make a pink, it needs to have the red and the white. So fill this in. And what are the chances of me getting a pink one? Just eyeball it. Type it in chat if you want.
All of them?
Yes, 100%. 4 out of 4, right? All of them are pink. So let's do a different scenario, right? Same incomplete dominance type, but I have a pink plus a white. What are the chances that they're gonna be white? So how many are white? I'm gonna go ahead and draw the square for you to be sure. So what are the chances that they're gonna be white? 50. Someone said 50. Let's take a look. I have 2 out of 4 here. That is 50%. Good job. So let's do this in a word problem this time. Same situation. If you are red, you have both of the red alleles. If you are white, you have both of the white alleles. And if you are pink, you have both of the pink alleles. If I combine a red plus pink, how many are going to be white? I'm gonna let y'all draw out the Punnett square this time. Oh, did I already get an answer? Dang, KJ, you big brained it. KJ said none. All right, can I get another answer out here? Just instantly do the answer. All right, I'm failing is agreeing with Luis, who also says none. So let's draw this out. I've got my square. I know red has to be double red and pink has to be red and white. Filling this in real fast gives me none. Fairly simple. All right, that is codominance and incomplete dominance done. Now we need to talk about lethal dominance. Lethal dominance means if you have two dominant alleles, that specific gamete will die. So dwarfism is the example she always uses, so dwarfism is going to be the example I'm going to use. If I have two heterozygous parents with dwarfism, meaning I have genes that look like this. Remember, heterozygous, I have one dominant, one recessive, one dominant, one recessive. So I draw a square that looks like this. So when we talk about lethal dominance, let's look at our ratios. 1 out of these 4 is going to die. It's this one. How do I know it's that one? I should do this in a different color. Give me one second. How do I know that one's going to die? Lethal dominance. That's how I know it's going to die. In lethal dominance, if you have both of the dominant alleles, you're dead. So I know 1 out of 4 of these is going to die. The other 3 are going to stay alive. So for the rest of these, I'm going to put these out of 3 rather than out of 4. So in this situation, how many are going to show dwarfism? Dwarfism is a dominant gene, which means 2 out of 3. Remember, because I've gotten rid of 1. 2 out of 3 are going to show dwarfism. And 1 out of 3 is going to not show dwarfism. So we're going to call them normal. Does this make sense?
Yes.
Yes. Okay, good. Remember, I am putting it out of 3 because I'm only considering the living ones. If she says them all, I'm pretty sure you put them out of 4. Because, yeah. So let's do a different one. Say, for instance, someone is, let's say you have a normal parent and a parent with dwarfism. What are the, give me all of the phenotypic ratios. Someone help out that person in the Discord server, please. They appear to be lost. Oh, it looks like I lost some participants partway through. Damn, that's sad. Give you a little more time. And then we will cover chi-squares. And hopefully I'll be done by 8:30. And I will get this video up at 9:30 because YouTube's slow sometimes. And I don't have the upload speed. Maybe next time I'll do this 2 days before so that I can get this, so I can get the recording up for everybody else. Do I have any answers yet? Remember, we are talking about lethal dominance. Do I have an answer? 50% for each. What is each? What traits am I considering for each? With dwarfism and normal? All right. Well, let's make our squares. So if we're talking about lethal dominance, I know that dwarfism is dominant, right? So a normal parent would have both recessive genes, and a parent with dwarfism must be heterozygous because any parent that is double dominant for dwarfism would technically be dead. So let's fill this in. I have 2 out of 4, or 50%, with dwarfism, and 2 out of 4, or 50%, who are normal. All right, that's it for this part. Let's go over chi-squares. So we're looking at chi-squares. Remember, it is the x squared equals the sum of the observed minus expected squared divided by the expected. Right, that's our formula. If you didn't already have it, well, now you have it. And so for this one, I don't know, what's a trait I can do? Hair type, right? So I'll do capital H as straight and lowercase h as curly. So say for instance, I have 2 parents, or sorry, my F1 generation is, what is it? Heterozygous plus heterozygous. And I observe from the offspring of this F1 generation, 47 with curly, 52 with straight. So let's do, I'm gonna erase the formula now. I hope all of you have it. So first thing I need to do is solve for my expected value, right? I need to know what to expect. Well, what's my total? 99. Why didn't I pick 100? Oh well, 99 total, right? I am measuring 99 offspring. And so, how do I find the expected? Well, I got to do some ratios. So I have a heterozygous plus a heterozygous. Let's draw that out. So, and remember, from this point on, we're going to think about phenotype, right? So 3 out of 4 are going to have straight hair. And 1 out of 4 are going to have curly hair, right? And so now I need to do this fraction that I have just found times the total. So someone give me some expected values. See what we expect. Why did I close my graphing calculator earlier? I knew I was going to do this. So that's 3/4 times 99 gives me 74.25 for straight. And remember, I don't round this part, I only round at the end. And what is that? 1/4 out of 99, 24.75. Did everybody get this? Yes?
Yes.
Yes, okay, good. So now Oh, I lost some more participants. That's sad. So now I do the fun part. I have to do the observed minus the expected squared divided by the my brain divided by the expected. So I have to do what? Observed minus expected. Let's do curly hair first. So what's my observed? Different color this time. What's my observed? 52. No, wait, 47 minus what's my expected? 24.75 squared divided by 24.75 plus new color. What's my observed for straight hair? Well, that's 52 minus my expected 74.25. Square all that, divide it by 74.25. And what does that equal? And round it to the nearest hundredths. Anybody? I know not all of you have graphing calculators, so I mean, I expect one of you to have it though. 26.7, nearest hundredths. So nearest hundredths, Jessica, so 2 decimal places. Yes, 26.67, that's what I got. All right, so now we get to do the fun part. Everybody have all this math. Everybody understand how I did all this math. This part y'all should understand the best, right? Even without me needing to explain it, this part should have been easy.
Yes.
Yes, okay, let's talk about conclusion statements. So now we know that this equals 26.67. I added twice because I was only looking at 2 things, right? So since I added twice, what is my degrees of freedom? Well, I take the number of times I added, aka 2 in this case, because I did all the numbers for curly and then all the numbers, or plus all the numbers for straight, and I take 1 away, because technically the first time you add, it is forced. So I added twice, I take 1 away, that means I have 1 degree of freedom. And then she's going to give you a table. You're going to look for the row or the column that says 1 degree of freedom. You are then going to find the number that's on the other column that says 0.05, and you are going to look for that number. That number is the critical value. And it is unfortunate that I don't have a table. I apologize profusely for that because I wasn't prepared. Anyway, you're going to look at the table, and let's say, for instance, this table is giving us these values, right? So we have, what is it, 0.05, and you have like 1 degree of freedom, 2 degrees of freedom, 3 degrees of freedom. And it's giving you like 1.2, 3.75, 5.76, right? And so if I look for the degrees of freedom, oh, wait, no, sorry, degrees of freedom is on the other end. And I look for this value on the side. Always look for the 0.05, unless she tells you otherwise. I'm looking at 1 degree of freedom. That means the number I'm comparing it to is 1.2. All right. So now I look. Is 26.67 less than or greater than 1.2? It is greater than 1.2, which means my chi-square number is greater than the critical value. And this allows me to conclude one of two things. If the chi-square, if x is greater than the critical value, then that means I have rejected the null. Which means all our results are not due random, or sorry, are not due to random chance. And that means there is something else influencing our results. And the p-value, you do the opposite sign, 0.5. So if I reject the null, what does that mean? It means my results are not random. They are statistically significant because there is something else influencing them, right? If this situation was reversed, if x squared is less than the critical value and p is then greater than 0.05, then I have failed to reject the null, and my results are random. If my results are random, that means they are not statistically significant and all of my calculations were correct, or all of my assumptions with the expected values were correct, right? So the easiest way to remember this is x is greater is reject, x is less is fail, and if you reject, it is not random, and if you failed, it is. I can't come up with an acronym on the spot. Just here, let me, if anybody wants to take a screenshot of this, give me 1 second. Let me match these same color. That is not magenta. Right? So remember, x is less, that means you failed. X is greater, that means you rejected, right? Everybody understand that. Have this on the side as like a cheat sheet for you, because this is probably going to be the part that stumps you the hardest. Everybody understand? Yes,
Yeah.
no, perhaps? Yes? Okay, good. Evolution is the last thing you need to cover, but evolution is really simple. It's just vocabulary. You should have that in your notes already. If you rewatch this video at least like 3 times, you should be able to memorize all the other stuff, and then you don't have to really memorize evolution. Other than that, that is it from me. Any other questions? Anybody? Anybody? No questions? All right. Well, if that is it, I need to do a generic YouTube video. Oh, wait a minute. I just got something in chat.
Wait, did she say that we had to have like our own chart, or is she gonna provide us with the chart?
No, she's going to give you a chart. Okay.
Right.
So how do you know what the degrees of freedom are? Okay, let me rewrite the equation here. So when I did the math, I did 47 minus 24.75 squared divided by 24.75 plus, is that 52 minus 75.25 squared divided by 75.25. Okay, So how do I know what the degree of freedom is? I added once, I added twice. That means I have added 2 times, I take away 1, that gives me 1 degree of freedom. Does that make sense? Okay. To further expand, if I added 1 more time, like say for instance, instead of just this, I did plus, I don't know, what's a random value? Plus 2 minus 25.2429 squared divided by 25.29. Let's change all the numbers on the bottom. If I added, for instance, that random number, right, I would, oh, whoops. I would now instead consider it 1, 2, 3. I added 3 times, I take away 1, which equals 2 degrees of freedom. That's how I find that. Usually your degrees of freedom will either be 1 if it is a dihybrid, or 1 if it is a monohybrid cross, or 3 if it is a dihybrid cross. But other than that, that's how you figure it out. And if what I just said confused you, ignore it. Just count how many times you added and take away 1. No questions? No more questions? Come on, guys. Right before the test. Review evolution by yourself, but right before the test is your last chance to ask questions. All right, I'm going to assume nobody has questions then. I need to do a generic YouTube outro, so say hi, YouTube, everybody, if y'all want to.
Hi, YouTube.
Hi,
Thank you.
YouTube.
Yo.
Hi.
All right, this is the end of the YouTube video. Don't forget to like and subscribe, please. I'm at like 10 subs. Can I get that to 11?
Say hi, YouTube.
Hi, YouTube.
Gender reveal at 20 subs.
Face reveal at 20 subs. Other than that, oh, everybody in here. Who's in here? Someone take a screenshot. And whoever I'm failing is, we're not going to count you for this one. Somebody take a screenshot of the participants list. If everybody on that participants list passes the test, I do the stream. All right. You better pass. There's even less people this time. Other than that,